Hi everyone. Today we are doing chapter 2, DNA biology, and I finally got my own new color printer, so I have your papers here in color, which makes me super happy. I'm also getting a new doc cam. I have it, I just need to figure out how to set it up so then it won't be upside down anymore. So goodbye to upside down Mikkel. Hello to your notes in color. Okay, sorry. A little excited here. Let's get down to the question of the day. What percentage of your DNA actually codes for proteins? So just make a guess in your head on this and we'll come back to that at the end. And for the pre-lecture assignment, let's just go through that quickly together. This chart should look somewhat familiar to you. We talked a little bit about it in our macromolecules chapter, I think it was. But pause this for a second and see if, well, this should already be done, but if you haven't, see if you can fill this in on your own without looking at anything. And then let's go ahead and fill it in together. So we're comparing DNA and RNA. We're going to talk about both today. So DNA has 2 strands. Oh, please don't make me go get a new pen in the middle of this. Two strands, while RNA only has 1. The sugar in DNA is called deoxyribose. And that's why we call it deoxyribonucleic acid, whereas the sugar backbone for RNA is ribose, which is why we call it ribonucleic acid. And then the bases are A, T, G, and C for DNA. And for RNA, they're the same, but we replace this T with a U. And that's just, those are representing the first letters of each of those bases. So the backbone, both of those, what they have in common is they both have a sugar phosphate backbone. But the sugar with DNA is deoxyribose. The sugar with RNA is ribose. Let's see. Yeah, we can go ahead and do this. So matching these up, ribosomal RNA, and it's often abbreviated like this with a little r than RNA, is B. It forms the small and large subunits of ribosomes. Messenger RNA is C. Think of it as the messenger that carries genetic information from DNA to the ribosomes in the nucleus, and DNA serves as a template for its formation. And then transfer RNA, or tRNA, and again messenger RNA, we use a little m in the front of it, and tRNA, we use a little t, is A. It transfers amino acids to ribosomes, and each carries only one type of amino acid, thereby there are 20 different types of these, since there are 20 different types of amino acids. And we might come back to those pictures in a little bit here. So DNA replication, we've already learned about this a little bit in the cell cycle, but remember that this happens during the S phase of the cell cycle. And if we look over here, we have some names for these. They're not too hard. But DNA is usually in that twisted ladder-shaped double helix, but we're straightening it out here to make it easier to see. So this would be our original double-stranded DNA, and we call this the template strand. So like it says there in your notes, this is the strand, the original strand that is being copied. And in blue here, you can see we've taken this side of the DNA, and here is that side of the original DNA here, and then this side ends up over here. So we kind of unzip this, open it up, and then we can make 2 new copies. We can match it up and have 2 identical copies here. And we'll learn about the process of that today. And these 2 new strands are called surprise daughter strands. And let's see here. I'm trying to see if I have a picture of what I'm trying to explain here. Actually, I do. Okay, so here is a close-up structure of DNA. Not in color, because this is before I had my fancy printer. So here's the DNA molecule kind of unwound, and you can see it's made up of a sugar phosphate backbone and then the base pairs. Yes, it's upside down on one side, then the other side looks normal. There's a reason for that, you don't need to know. But let's look at this side. So we've got the sugar phosphate backbone here, and then the nucleotide, or the base is what we'll call it. So these bonds through here that you're seeing are hydrogen bonds, and if you remember back to our bonds, we learned about ionic, covalent, and hydrogen bonds, and hydrogen bonds are the weakest of the 2, or the 3, so it's not super hard. It doesn't take a lot of energy to unzip this because they're held together with those hydrogen bonds. So we unzip this, add the matching base pairs, and now you have 2 identical copies. I don't know if it says this anywhere in the notes. I don't think it does, but you need to know that the base pair A will always go with the base pair T, and the base C will always go with G. And the way I was taught to remember this is apple tree, A and T, and then chewing gum. C and G. So those base pairs always go together. You're not going to get a T matching up with a C or a G matching up with an A. And let's talk about some of these enzymes that are that play a role in this DNA replication. So if something ends in ASE, remember it's an enzyme. And let's skip down to DNA helicase. So if you just look at that word, it ends in ase, so we know it's an enzyme, and then we have helic in front of it, so think of that as helix. And so what this does is it breaks the DNA double helix. And so DNA helicase is what's going to come in and unzip these 2 strands so they can be replicated. So breaks the double DNA helix, it unzips the DNA in preparation for a copy to be made, and then the nucleotide bases which are our A's, T's, G's, and C's, are added to match the template strand. So we are left with 2 identical copies. Shoot, I was looking at the wrong place. And I've probably really confused you. This goes up here. So we'll come back to that. What should have been written here is it breaks the hydrogen bonds between the nucleotide bases to unzip the helix. I thought that sounded funny. If you have an eraser, erase that and fix it. Breaks the hydrogen bonds between nucleotide bases to unzip the helix. And that's going to go here. So swap those. Sorry about that. All right, now let's look at DNA polymerase. So it ends in ase, and the word before it is polymer. So if you remember polymers, if you put a bunch of the monomers together, you get a polymer, and this makes DNA polymers. So this is where it's going to add the nucleotide bases A, T, C, and G to match the template strand. So this is what actually builds the daughter strands. All right, and for each of these processes, going back up here to where we started at DNA replication, you need to know what you're starting with and what you end with. So with DNA replication, we start with DNA. And well, you're replicating it, so what do you end with? You end up with DNA. That one's pretty easy. So let me just clarify one more time. DNA replication happens during the S phase of the cell cycle. The reasons you would need to replicate your DNA are for mitosis or meiosis. And the enzyme DNA helicase unzips the template strand so that DNA polymerase can come in and build the missing strand. And then you're left with those two daughter strands. Mutations. We learned about these already, but they're any heritable change in the base sequence of DNA. So this can occur during replication, this process that we just talked about. It's not a perfect process. Mistakes can be made. And we also know that mutations, in our cancer chapter, we learned how they can be caused by like radiation or chemicals or carcinogens. And mutations, we kind of always think of them as a bad thing, but it's actually another source of genetic variation. Which is a good thing. Because the more variety you have in a species, the more likely they are to survive. And we'll get into more of that in our evolution unit. So we have a lesson that I teach to the 7th graders. It's called Harmful, Helpful, or Neither, and it's talking about mutations. And so mutations aren't always bad. So we have seen cases where they can be harmful and they can lead to these genetic disorders or to cancers. They can be helpful. So if you have a bug that is yellow and it lives in a green leafy environment and it gets a random mutation that just happens to code for it having a little bit more green to its color. Did I say skin? To its color. That's going to be an advantage because it's going to blend in, be less likely to be seen by predators and eaten. So that would be an example of a good mutation. And then they can be neither. Depending where the mutations happen, sometimes it doesn't even cause a change in the protein that is made. And sometimes it's in a section of the DNA that doesn't even code for proteins. And sometimes it can lead to a mutation that is neither good nor bad. So anyway, it can be helpful or neutral. But usually harmful. And then these mutations are passed to offspring. And again, that's how we get evolution. So that slightly more green bug is going to be more likely to survive and reproduce and pass on its green colored mutation to its little bug babies, and then in turn they are more likely to survive and pass on that gene, and you should see an increase in little green bugs or greener bugs in that population. But we'll get to that later. I'm sorry, I love evolution, so I'm getting ahead of myself here. But okay. On to the next page. Gene expression. This is something I make my 7th graders memorize as well, this order here. So DNA gets transcribed into RNA. The RNA gets translated into a protein, and those proteins code for traits. So let's write that down. What happens at this arrow is transcription, and we're going to talk about these processes. And then at this arrow here, we have translation. So if we look at our picture here, transcription happens in the nucleus where the DNA is, and it forms our strand of RNA, which then leaves the nucleus, and then translation happens outside here in the cytoplasm with using ribosomes and transfer RNA. So again, I'm kind of jumping ahead, but we'll get there. So DNA is transcribed into RNA. And so if you think of transcribing something, like if I were talking right now, and the recording software I was using was transcribing my words into like captions that you could read, think of transcribing like that. You're just kind of changing it to a slightly different version that is accessible to other things. It's a horrible explanation, but it'll make sense. And then translation, if you take what I'm saying and translate it to a completely different language, that's you're going to end up with a more drastic result. I don't know if that helps you at all or not, but when we talk about this, you'll see. So RNA polymerase, just like DNA polymerase, makes RNA polymers. And in a DNA sequence, with transcription, if we had a sequence of DNA, and I'm writing it in triplets for a reason that we'll see in a little bit. Let's say we had a sequence of DNA that looked like this. And we transcribed it. It would end up being U, U, A, C, G, U, A, G, U. Okay, so remember, A always goes with T, G always goes with C, but there are no T's in RNA, so anything that would have been a T is now going to be a U. Okay? So transcription, let's get into this. The process of making M, or messenger RNA, from a DNA template. That's kind of what I just showed up here. So the gene being copied unzips to allow access to DNA. Okay, so with DNA replication, we unzip all of it and copy all of it. But with transcription, you're only going to unzip the gene that codes for the protein that you need. Otherwise, unzipping the whole thing would be a waste of time and energy and resources. So it only unzips the portion of DNA that is needed. And then the complementary RNA bases are matched to bases on the DNA strand. Like I did up here. So let's say this is our gene. So we're going to unzip just this little area of the DNA. And then messenger RNA is going to end up looking like this. And just pair up with that section right there. So the mRNA leaves the nucleus through those nuclear pores that we saw in the nuclear envelope. Leaves the nucleus, and then the DNA, we're done with that part for now, so it zips back up until that gene is needed again. Alright, so transcription, what goes into that is DNA. We start with DNA, and our ending product is messenger RNA. Next page, a codon is a sequence of 3 nucleotide bases, and each codon codes for a specific amino acids. So if you remember our macromolecules unit, the monomer amino acids, when you put a bunch of those together, what's the polymer that makes? Take a minute, see if you can remember. It makes a protein. Okay, so if this were our RNA sequence right here, we've got these triplet codons. So we've got AUG, GUC, and so on and so forth. And each of those 3 letters codes for a specific amino acid. And then there are also certain codons that code for stopping the process, like to stop adding amino acids. But yes, just know that in RNA, we break these down into triplets codons, and each of those codons codes for a specific amino acid. We'll come back to anticodons in just a second, but we can fill this one in. There are 20 different amino acids. So the genetic code, the mRNA strand directs the sequence of amino acids based on those codons. So the start codon, and we're really simplifying this for you guys, it tells ribosomes where to start translating. And there's one start codon, and that is AUG. So over here, this shows all of your codons and the amino acids that they code for. And let's see, where is it? Here we have AUG is the start codon. We have stop codons as well. So stop codons tells the ribosome where to stop translating, which makes sense. And there are 3 stop codons, UAA, UGA, and UAG. You don't need to memorize those, just know that there are start codons that tell the ribosomes where to start adding amino acids, and there are stop codons that tell them where to stop. Okay, we're still going to come back to anticodon in just a minute. We're going to move on to translation for now. So translation now is the process of using mRNA to assemble strings of amino acids. Alright, so with translation, we're starting with mRNA. And we're ending up with strings of amino acids, which are essentially proteins, before they've been all folded up into their secondary, tertiary, and quaternary structures. So translation happens in the ribosome. Remember those organelles that we talked about? And the ribosomes look kind of like, I've heard it described as a hamburger. So here is a ribosome. It's got the top and then the bottom, like the larger bun on the top, and then the bottom down here. And in the center, it kind of sandwiches in this mRNA. So the ribosome finds the start codon and sandwiches the mRNA there. And then it takes the amino acids from transfer RNA and links them together into a polypeptide. Alright, so tRNA. What does transfer RNA do? It reads those mRNA codons. So those triplet codons. And it uses anticodons to insert the correct amino acid. And we'll get to anticodons right after this. So each tRNA is made to carry only one specific amino acid. And how many types of amino acids do we have? We have 20, which means there are 20 types of tRNA. Alright, so tRNA is this cute guy right here. And it has the anticodon here. So you've got the mRNA coming through the ribosome here. And then the transfer RNA is going to come match up at this site, and it has to have a certain anticodon to match up. So we got G and C, U goes with A goes with U. And so let's go back to our anticodons before we talk more about that. So anticodons are a sequence of 3 nucleotide bases that are complementary to the nucleotide bases of a codon. So over here, we've got a less cute picture of the tRNA. And here is the messenger RNA that would be going through the ribosome. And our codon here is UCG. And so the transfer RNA has to have the complementary strand to that, otherwise it can't fit. Alright, so the anticodon for UCG would be AGC. Alright, so if we look at this, remember there are 20 amino acids. Proteins are these chains of amino acids like wound up together. And you've got the transfer RNA, it's got the anticodon, it's got a specific amino acid that goes with that. And so it will come and bind to the codon that is complementary to it, kind of drop off its amino acid, and then the ribosome will attach those amino acids together. And I don't think you necessarily need to know this, but the top part of the hamburger is called the large ribosomal subunit, and the bottom bun is called the small ribosomal subunit because one is large and one is small. And then here in this image, you've got the mRNA going through here, and then at the same time, it's building this polypeptide, the chains of amino acids. So this is like more like a real life picture. And then this is a picture showing all the codons and the anticodons and the name of the amino acids and so forth. So what I would suggest doing, just so you can kind of watch this process, is I would go to YouTube and just type in protein synthesis, and there are tons of good videos that will actually walk you through and show you the process of transcription, translation, and how this all actually happens in the cell. So I would recommend doing that to kind of cement all of this in your mind. And then back to the question of the day. What percentage of your DNA actually codes for proteins? So think of whatever you guessed. And the answer is only 1%. So all that DNA you have in there, only 1% of that actually codes for proteins. 75% is for genes to kind of help with what these proteins do, or I'm not exactly sure, but the other 75% is used. So the other 24%, what the heck is that for? We don't know. As far as we know, it doesn't do anything, but I have a hard time with that answer. But yeah, the scientific community has not made a conclusion, I don't think, as of now, as to what the remaining 24% does. But yeah, you need to know this number, that only 1% of your DNA actually codes for proteins. Alright. This is optional, but I got through that pretty quick, so we're going to look at it. So we've got one strand of DNA here, and we're going to do a no mistakes replication and see what we get here. And then this side, we're going to make a mistake. So if no mistakes were made in this replication, rewrite the above DNA sequence below. Is it wanting us to do the complementary strand? I think it wants us to do, I'm assuming it wants us to do a complementary strand because this is in replication. So remember, A goes with T, G goes with C. So C would have a G here, G would have a C, T would have an A, T, A, G, 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 C, T, T. So practice filling that in, see if you can do it on your own. And next, the DNA was transcribed into mRNA, so write the complementary mRNA strand below. So remember, there are no T's. So C, G, G, C, A. Instead of putting T's here, we gotta put U's, because this is RNA. A, T, A, G, C. I got off somewhere. Let's see. C, G. I'll equal for C, G, A. Alright, so we just replicated that DNA, and then we transcribed it. Now it's mRNA, and now we're going to translate it using this very cute, I love this, Guide to the 20 Amino Acids. You can use this, or I think it's easier to use this table right here. So let's sketch out our codons. So 3 base pairs per codon. And then we can see, using this table, what they code for. I know mine is tiny, so, well, let's see, our first one is CGU. So there's an easy way to use these tables. It gives you the line for the first base. So then that'll narrow it down to one of these 3 sections. And then the second base will narrow it down even further. And then the third base will give you what you're looking for. So we're looking for C. So that's going to be somewhere in here. And then G is going to be somewhere in here. And then U, so this is arginine. Okay, and our next codon is UAU. So UAU, that's tyrosine. And then next up we have CCC. What a fun one. CCC, that's proline. And our last one is GAA. So GAA, glutamic acid. Alright, so there's our sequence of amino acids. And so I'm not going to do this part. I want you guys to do this on your own. So on this left-hand side, we just did together what would happen if there were no mistakes and what our resulting amino acid sequence would be. And on this one, you're going to make a mistake. So you can decide, do you want to delete a nucleotide? So you can completely cross one of these out during replication, it just disappears. You can insert an extra one, so you'd have an extra box here. You can switch places with two neighboring nucleotides, like you could swap those two, or sometimes you can even take like a whole sequence and like rotate it. So just make a mistake when you're copying, and then transcribe it into mRNA, and then translate it into the amino acid sequence. And then notice these questions. Did your amino acid sequence change after the mutation? Sometimes it won't. If it did, what are the possible consequences of this mutation? And this is just a random, we're not even telling you what gene this is for or what protein it codes for. So just think of some of the possible things that could happen because of that mutation. So that is it. Again, make sure you know what goes into each process. So DNA synthesis, we start with DNA, we end with DNA, or DNA replication. With transcription, we start with DNA that we unzip, and we make a little piece of mRNA. That's what we end with. And then that mRNA leaves the nucleus, goes into the cytoplasm to the ribosome, and is transcribed with the help of tRNA and the ribosome. And the tRNA has those anticodons that match up and drop off their amino acids, which get chained together to make a protein. Whew, that was a quick little summary there. And again, I would recommend looking up a video to watch the actual process of this. That would be very helpful. So I can only do so much with paper and pen. All right. Have a great day, guys.